I'm Eddie Watkins, a professor of biology at Colgate University in upstate New York. And today I want to demonstrate to you sort of the use of one of our principal tools that we use for ecophysiological measurements. This machine is the LiCor 6400 XT system. It is a system, essentially a field portable infrared gas analyzer. And it's the machine that we use to measure a number of aspects of plant physiology, principally being that of photosynthesis. We also can measure respiration. We can also measure conductance, the leaf conductance to water vapor, to understand how water moves out of the leaf. So with additional tools that I'll demonstrate in just a moment, we can actually measure simultaneous processes that are occurring in the light reactions and in the dark reactions. And I'll be able to put a leaf into the machine and quantify not only sort of the rate of photosynthesis, but also the rate of electron transport or electron flow into the photosystems. Now the machine is it's highly sophisticated, and it allows us to really measure things very precisely in these plants. Naturally, I use it to study ferns, and we have a lot of questions that we're going to learn more about in the future blog, but I'll demonstrate how this machine works, because it really is the first sort of field portable infrared gas analyzer system that is survivable out here in the tropics. So with this machine, then again, we can control many different variables. Important as which, of course, water vapor. We can remove water, or we can add water to the machine by using this desiccant scrubber. We can also take out CO2 or add more CO2 from the atmosphere by using this CO2 scrubber that's full of soda lime. And then importantly, we can precisely control the amount of CO2 that's entering the system by this special CO2 mixer on the side. And underneath this steel column here, this tube, there's a standard CO2 cartridge that you would use in a nail gun or in a pellet gun, BB gun. And we're actually going to be able to tell the machine to supply a very specific amount of CO2 into our leaf, and that will allow us to control the aspects of CO2 or change CO2 if we wanted to, if we were asking questions about how CO2 changes influence photosynthetic rate, etc. So apart from measuring CO2 with this mixer, we can also make very detailed measurements of light or control the amount of light that's arriving to the leaf. For example, if I turn on the light inside the chamber here, I'll turn it on, and now I'll open it up, and you'll see exactly how much light is coming in. I've just set this to 3,000 micromoles per meter square per second of light. That's essentially full sun. I have really nice fine scale control. For example, if I turn this down to 200, or let me turn it down to 300, so that is order of magnitude lower, you see the light goes down. I can also turn it down very low to 1 micromole, which is almost undetectable by the human eye. So we have a lot of fine scale control over the light that we can provide to a leaf. And that's important in understanding sort of the maximum function of the leaf. So if we want to understand the maximum photosynthetic rate of a leaf, then we have to be able to change light in a way that will allow us to get to that level. We can also measure, by turning the light off, we can also measure the maximum respiration rate. So we put the leaf in there, we let the leaf stay in dark for a long period of time, and we can get the maximum respiration rate. So let me now demonstrate how you do this with a fern. One of the projects we're working on are the blue ferns. You'll hear more about this in a future blog, but this is one of the blue ferns. And what I'm going to do is I'm going to run a photosynthetic induction curve on this plant to show you, to understand how quickly this plant goes from basically a very low photosynthetic rate to its maximum photosynthetic rate at a given light reading. So I'm going to open up the chamber, and I'm going to turn this on to 2,000 micromoles of light, which for this fern is a very typical amount of light that it might see. This is a high canopy epiphyte. I'm going to put it in the chamber, and I'm going to close the chamber on it. Now it's completely sealed inside this chamber. And what's happening now is we're supplying a very specific amount of CO2. The CO2 is going in one tube. It goes into the leaf. If the leaf is functioning, the stomata are open. It takes in CO2. It also releases water vapor into the airstream. That then goes through another infrared gas analyzer, is mixed up, and the machine then does a calculation based on knowing what's going in and what's being taken up, and it calculates things, or it models things like photosynthesis, stomatoconductance, and a number of other variables. So we'll leave this thing running for a few minutes, and we'll run an automatic program that I've written to look at the photosynthetic induction. So now this program that I wrote has been running for about 20 minutes, and what we see on this graph I've pulled up on the machine, we have essentially graphing two functions. The solid squares represent stomatoconductance, and the pluses represent photosynthesis. So this axis is photosynthesis, here's conductance, and on this axis is time. So what you see is that in the beginning in terms of photosynthetic rate, we established a flat line. We know what the sort of normal photosynthetic rate of the plant is at this relatively low light. We turn on the light here to 2000 and the plant slowly begins to climb up, and eventually if we run this a little bit longer it will saturate. And the same thing is happening here. We have established a baseline here, and as we turn on the light, the stomata conductance increases. So the stomata open up to take up more CO2 and to cope with the excess light that they're receiving. So that's been a quick demonstration of how I use the LiCor 6400 in my research to study plant physiology, and I hope it leaves you with a better understanding of how plant physiologists approach the study of plant physiology and how we use this piece of equipment in the field.